Hi guys, welcome to Raaonline. So today we are going to deal about a biochemical test which is going to be intended for the MD DNB postgraduate in exam point of view. So it is going to be a very short session, I will not take much time, and we will see about this particular test in a gist. So here we are going to use the phenylalanine agar, and we are going to study if the particular organism is able to produce the enzyme deaminase. So in case if the particular organism is going to produce the enzyme deaminase, what is going to happen is that we are going to use the phenylalanine agar, which is going to contain phenylalanine. So in case of the particular organism or bacteria produces the enzyme deaminase, and the phenylalanine will undergo the oxidative deamination with the help of the deaminase, which is produced by certain organism which is belonging to the proteae like Proteus and Morganella, and the particular phenylalanine is going to get converted into phenyl pyruvic acid. Phenyl pyruvic acid, again we are going to find out by adding a reagent. So here the particular reagent which you are going to use is going to be freshly prepared 10% ferric chloride. So the phenyl pyruvic acid which is going to get formed in the first step. So the particular phenyl pyruvic acid is going to react with the reagent that is going to be the 10% freshly prepared ferric chloride. So it is going to react with the ferric ions of ferric chloride to form the greenish black color. In case if you are going to see this particular tube, so this is the first tube and this is going to be the second tube. So second tube there is no color change. So which is indicating a negative test. And this one is having a greenish black color. So it's going to be positive test. Here we are going to use a 10% freshly prepared ferric chloride. So the particular phenyl pyruvic acid which have been formed by the deaminase enzyme. So the particular organism is going to produce a deaminase which is going to convert the phenylalanine into phenyl pyruvic acid, which is called as oxidative deamination, and the particular phenyl pyruvic acid which gets formed, so it is going to react with the freshly prepared 10% ferric chloride. So there is going to be the presence of ferric ions in the ferric chloride. So the particular organism is going to produce the enzyme deaminase, and there is going to be conversion of phenylalanine to phenyl pyruvic acid, which is going to react with the ferric ions of freshly prepared 10% ferric chloride to form the greenish black color. So why we are going to do this test? So in case if you are going to get anything like a non-lactose fermenter, we need to know whether it's going to belong to the organisms like Proteus or Morganella. So organism belonging to the triproteae like Proteus, various Proteus like Proteus mirabilis or Proteus vulgaris, Proteus penneri, and we have something called Morganella morgani. So these are the various organisms which is going to be PPA positive. So in order to differentiate them, we are going to use it. And the particular quality control intended for the PPA test, the positive control is going to be the Proteus species and the negative control is going to be the Escherichia coli. So what is the procedure? So here we are going to take a colony from the agar and we are going to inoculate into the PPA agar plant which is going to contain the phenylalanine, and we are going to incubate for 18 to 24 hours. After incubation, we are going to take this plant out and add 10% freshly prepared ferric chloride which is going to contain the ferric ions, and the formation of green-black color complex which is going to indicate the positive test. So in case of positive test, it can be the organism belonging to the Proteus like Proteus mirabilis or Proteus vulgaris, Proteus penneri, or anything like Morganella which is going to turn out positive. In case of any organisms like Klebsiella, E. coli, or something like Enterobacter which is going to come negative for PPA test. So this is about today's biochemical. So I hope you guys have an idea about the PPA test and some other biochemical in the upcoming session. I will catch you guys later. Till then, Dr. Ashok signing off.